Okay, well, thank you to everyone at Bionano for inviting me. My name is Darren Finlay. I go by my nickname, Ben. I'm from Sanford, Burnham, Prebys Medical Discovery Institute in La Jolla, California. And today I'm going to be talking about data from our recent paper. So 70% of what I'm going to be talking about today is from our recent paper from the molecular therapy family of journals. And it was entitled Unveiling Genomic Rearrangements in Engineered Induced Pluripotent Stem Cell Lines by Optical Genome Mapping. You can download it here from the QR code, or you can just Google my name and I'll show the QR code again at the end. But the caveat I want to get in here is that optical genome mapping is for research use only. And the only thing we used it for was research. This study, or the samples used in the study I'm going to show today, are a collaboration with the Kaufman Lab at the Moores Cancer Center at UCSD. And each of the samples we're analyzed by optical genome mapping were just experimental model systems. They were never intended for clinical use. So a quick little bit of background into CAR-NK, or natural killer cells. This is from a Kaufman Lab review in 2009. I think it's well established now that pluripotent stem cells can be generated into hematopoietic precursors, which can then be induced to form any type of blood type cell, or indeed any other type of cell in the body, or almost any other cell in the body. But Dan's group are interested in natural killer cells. So NK cells can be differentiated from iPSCs. They have some advantage over CAR T cells in that they don't require autologous collection and matching, and they have a lower endogenous toxicity and can recognize tumors without human leukocyte antigen, or HLA. So to make natural killer cells even more efficient killer cells, they're modified with chimeric antigen receptors, or CARs. And this is just a graphic example of a type of CAR that would be put into a natural killer cell. So one takes one's iPSCs, expresses your CAR or whatever marker gene of interest, and you differentiate them into NKs, and then not the samples I'm going to be showing today, but these can be used then for targeted therapy for cancer. But obviously during the modification of the cells or during the culture, inadvertent genomic alterations can occur. So just to make everything more awkward, I'm sure everyone has seen enough circus plots. I want you to go through that, but we do a different color scheme. So deletions are in bright red, insertions are burgundy, inversions are lime green, duplications are blue, and translocations are in this teal color here. But spoiler alert, because we're inserting material into cells, you're going to be seeing a lot of these burgundy dots. So this here is a karyotyping analysis of one of our parental or starting iPSCs. So these are supposedly healthy cells, and this is just the metaphase spread that was carried out by a contract research organization on these cells. And they reported back that it's 46 chromosomes, XY, normal human male karyotype. Nothing of any interest according to karyotyping. However, by optical genome mapping and analysis through the rare variant analysis pipeline, or RVA pipeline, we actually detect in this same sample 6 deletions in red, 2 insertions in burgundy, an inversion again in lime green, and then duplications in blue. But we also see copy number loss all around the genome and these little red peaks on the gene track. And the caveat I want to say is that we use the rare variant analysis, so we filter out any structural variants that have ever been detected in a healthy human donor. And again, we only show we only filter out to only show structural variants that are within 12 kilobase pairs of a canonical gene. So there's there are a lot more structural variants here that aren't being shown on the circus plot. This is the same image from the previous slide, but in this case, all of the structural variants have also been plotted on this graph, and based on their size and their VAF, or variant allele frequency, which can be considered a measure of clonality. So all these deletions and insertion here have a variant allele frequency between 0.4 and 0.6, which means that these are probably heterozygotic events in all of the cells. At the bottom is the copy number image that is produced by the Bionano software, and every red line here represents copy number loss, as we saw on the gene track here, and we can see that there's copy number loss all throughout the genome. Example of what some of these structural variants can be, this duplication here is in a gene called NBPF6, or neuroblastoma breakpoint family. And neuroblastoma is a pediatric cancer. It's a pediatric neural crest cancer. And that's where this breakpoint or this gene was first identified with this sort of rearrangement. But more recently, we have observed this exact same rearrangement in many lymphoma samples. So maybe if I behave myself, Bionano will invite me back next year to tell that story. And here is the gene level map. Optical genome map is always blue or sapphire. And what we can see is the optical genome map maps perfectly along through the chromosome, all the way through NBPF6, and then we get this inverse duplication event here. So I would suggest that this cancer-associated event is probably not something you want in your parental iPSC cells. Another example, again, of our parental cells from a healthy donor. This, again, done by a contract research organization. This is from their report. This is the karyotyping showing 46 chromosomes, XX, normal human female karyotype. Nothing wrong, perfect. However, when we run the rare variant analysis after optical genome mapping, we find that this sample actually has 3 deletions, 14 insertions, an inversion, and copy number gain all around the genome. The inversion event is 150 kb, and the copy number gain on chromosome 1 is 4 megabase pairs, which is why you can actually see it there. It's so big. And then this is again the structural variance plotted on the basis of size versus variant allele frequency. So what we can see is these insertions and this deletion are in around 0.5, so probably heterozygotic events in all the cells. But we also see a smattering of these insertions, all of the same size at various different allele frequencies. So this suggests that this supposedly normal iPSC had been genetically modified previously, but none of these insertions were noticed by karyotyping because they're 6,000 base pairs and they're below the threshold of detection through traditional karyotyping techniques. And then at the bottom, it's just the gene copy number. Again, in this case, the software calls gains as blue and losses as red, and we can see lots of gains and some losses throughout the whole genome. So far, we've seen that optical genome mapping through the rare variant analysis pipeline can detect structural variants in parental Cs that were previously deemed normal. Now we're going to analyze cells that have been genetically modified, which could, of course, result in off-wanted or unwanted off-target effects. And from here on in, we're going to be using the dual analysis feature to compare the parental cells with the progeny or the engineered cells. So only structural variants unique to the engineered cells will be shown. And for the examples today, we use 3 different methodologies. We use lentivirus, transposons, or CRISPR-Cas safe harbor insertion. It's just a little bit of intro to the CRISPR-Cas safe harbor insertion technology. This graphic I just took from System Bio's website. So the safe harbor site where people target is a known insertion site for adeno-associated virus. But in this technique, no virus is involved. CRISPR-Cas is used to make a deletion in the same safe harbor site, which is actually this gene called P1 or 12C, but no virus is involved. Here is an example of an iPSC cell that has been developed using this technique. In this case, they were trying to induce mesothelin and also brain angiogenesis inhibitor factor. It's not so relevant for our talk. And what we see is there's only one structural variant detected. And it's an insertion, and it's in chromosome 19, exactly where we would expect it. And here is the gene level map showing optical genome mapping in blue, and the reference sequence chromosome 19 in green, and we can see this insertion of around 7 kb right here in the gene of interest. This is another example where they were trying to express CD123 and a secreted version of IL-15 because it helps these cells survive in vivo. And again, we see the correct insertion in chromosome 19, but we also unexpectedly find a duplication of this gene EMSY or MC. This is the gene level map here. The optical genome map maps correctly through the gene, and then we get this, again, inverted duplication of EMSY or MC. And this gene has been linked to sporadic breast and ovarian cancer. So again, I would suggest not a structural variant you want to see in your cell population of interest. So we also used some cells that had been modified using transposons. So transposons were a big thing many years ago. They've somewhat been overtaken by CRISPR-Cas9. And transposons, sorry, I just took this graphic from Adgene's website. They will sell you this technology if you want. But transposons target TTAA tetramers, which, as one can imagine, are interspersed throughout a 3.2 gigabase pair genome. And insertion efficiency is controlled by using different concentrations of DNA and enzyme or transposase. And these are just 3 examples of iPSCs that were edited using transposon, in this case, the piggyback system. This sample here has 19 insertions. And if we plot the size versus variant allele frequency, we find that all 19 are pretty much the same size, except maybe this one. And they're spread throughout the genome, and they all have a variant allele frequency of around 0.5, between 0.4 and 0.6, suggesting that this is a clonal population of cells with 19 different insertions throughout the genome. So way too many for an iPSC, or a genetically modified iPSC. This example, we have 8 insertions, but also 3 deletions. And again, this is the size versus VAF plot here. You can't see the deletions because they're much bigger than 20 kilobase pairs. And then a final example, it was this sample here, where we saw 46 insertions, a deletion, an inversion, and even a translocation between these 2 genes, AH1 and SYNE1, which is predicted to form a fusion protein. We look at the size versus variant allele frequency graph. We see lots of them are the predicted size, around 7,000 base pairs. And again, variant allele frequency around 0.5, but we find other insertions of various different sizes. And this is the gene level map of the translocation predicted fusion, where the optical genome maps through AH1, forms a fusion with SYNE1, and then carries on through this gene, which is actually estrogen receptor 1. So probably not a region of cells you want to mess up through editing. Which brings us to lentivirus. So probably everyone has heard of lentivirus or lentiviral modification of cells. Just a graphic I took from Miltenyi's website. But lentivirus are a subset of retrovirus, the most famous of which is HIV. Of course, lentivirus in the lab are modified, so they're not replicative, and they have some advantages over the other techniques in that they can carry larger cargoes of DNA, but they do insert somewhat randomly into the genome. It's hard to control where the insertion occurs. And this here are just 2 examples of cells that were genetically engineered using lentiviral engineering methodologies. And I remember this one specifically because when I first saw it, I thought, okay, I've messed up. I've obviously not set the filters correctly. But I went and checked everything and it seemed correct. So I reached out to Ben Clifford at Bionano and asked him to go over my filters and my settings. And he said, no, these all look fine. So this sample genuinely has 105 insertions and 2 deletions. And most of these insertions appear to be of the predicted size, around 7 kb, although there are 2 that seem to be twice the size. We've never actually sequenced to find out what they were. And so, of course, 105 deletions being way more copies than you actually want in your cells. This sample here only has 51 insertions and also has a deletion in the gene PLLP. And this is just the gene level map showing that deletion there. And again, we're only showing structural variants that are within 12 kb of a canonical gene. So there actually are more structural variants in these samples than we are showing on the Circos plots. Then a final example was of a deletion. So often for these NK cells, one wants to delete the gene or impair the gene CISH, or cytokine-inducible SH2 containing protein. And in this instance, we make a 60 base pair deletion within the CISH gene to impair it. And this is the optical genome map here, which doesn't detect it because 60 base pairs is way below the detection threshold for OGM. However, the deletion is there. It's been confirmed by PCR. But somehow, during the modification, making this deletion, we also detect two deletions. One is quite large, has 4 genes in it, but there's also a deletion in the gene called EF2K, or eukaryotic elongation factor 2, or CAMK3. And both these deletions, again, seem to be heterozygotic in all the cells. And here's the gene level map showing that approximately 50 kb deletion in this essential gene. So in summary, hopefully I've been able to show that optical genome mapping unfortunately detects structural variants in purportedly normal iPSCs. The dual analysis feature allows us to directly compare the engineered and parental cells to find hopefully the expected SVs, but also off target. The CRISPR-Cas safe harbor insertion seems to be most precise, whereas lentiviral and transposon technologies show excess insertions of cargo. But all 3 methodologies can result in off-target genomic events, including in genes of potential clinical significance. Based on all of this, I would suggest that OGM could be a valuable QC step in genetic engineering techniques. It's just by way of acknowledgement, my mentor, Christina Vawori at Sanford, Burnham, Prebys. Nothing I do would be possible without her. Rabi Murad is our director of bioinformatics, who assisted with analysis and figure preparation. And all the samples are from Dan Kaufman's group at UCSD. Pooja Hoare was my co-author on the paper, and some of the other samples were made by Ben Goldenson and Zhao Ali. And as I mentioned before, invaluable help from Ben Clifford and everyone at Bionano Genomics. Thanks.